6th Developmental Biology Long Question. 1. What is gametogenesis? Describe about the process of spermatogenesis. 6 marks. Answer. Gametogenesis. Gametogenesis is the biological process of forming haploid gametes, sperm and egg cells, from diploid germ cells through meiosis. It occurs in the gonads, testes in males, spermatogenesis, and ovaries in females, oogenesis. Spermatogenesis. Spermatogenesis is the process of sperm cell formation in the seminiferous tubules of the testes. It involves 3 main phases. Multiplication phase. Diploid spermatogonia undergo repeated mitotic divisions to increase in number. Some spermatogonia differentiate into primary spermatocytes. Growth phase. Primary spermatocytes grow in size without dividing. Maturation phase. Primary spermatocytes, 2N, undergo meiosis I, forming 2 haploid secondary spermatocytes, N. Secondary spermatocytes undergo meiosis II, producing 4 haploid spermatids, N. Spermiogenesis. Spermatids mature into spermatozoa, sperms, by developing a tail and acrosome. Spermiation. Fully developed spermatozoa are released into the lumen of seminiferous tubules. This entire process takes about 64 days in humans and ensures continuous sperm production throughout life. 2. Give account of phases of development. 6 marks. Answer. Phases of development. The development of an organism from a single-celled zygote to a fully formed individual occurs in several distinct phases. The major phases of development are: gametogenesis. Formation of haploid gametes, sperm and egg, through meiosis in the gonads. Involves spermatogenesis, in males, and oogenesis, in females. Fertilization. Fusion of the male and female gametes to form a diploid zygote. Restores the chromosome number and activates embryonic development. Cleavage. Rapid mitotic divisions of the zygote without growth, forming a multicellular structure called the morula. Cells formed are called blastomeres. Blastulation. The morula develops into a blastula, a hollow ball of cells with a fluid-filled cavity called the blastocele. Gastrulation. Rearrangement of blastular cells to form the gastrula, which has 3 germ layers. Ectoderm, forms skin, nervous system. Mesoderm, forms muscles, bones, circulatory system. Endoderm, forms digestive and respiratory tracts. Organogenesis. Formation of organs and organ systems from the 3 germ layers. Examples: neural tube forms the brain and spinal cord, somates form muscles and bones. Growth and differentiation. Cells grow in size and specialize into different tissues and organs. Leads to the formation of a fully developed embryo. Metamorphosis, in some organisms. In amphibians, e.g., frogs, transformation from larva, tadpole, to adult. This systematic development ensures the proper formation of a functional organism. 3. Give an account of development of frog up to gastrulation. Answer. Development of frog up to gastrulation. The development of a frog, e.g., Rana tigrina, follows a systematic process starting from fertilization to gastrulation. Fertilization. External fertilization occurs in water. The sperm fuses with the egg, forming a diploid zygote. The zygote has animal and vegetal poles. Cleavage. The zygote undergoes rapid mitotic divisions, cleavage, without growth. It is holoblastic, complete, and unequal due to yolk distribution. Forms a morula, a solid ball of cells, blastomeres. Blastulation. The morula develops into a blastula, a hollow structure with a fluid-filled cavity called the blastocele. The blastocele is more prominent in the animal pole due to the presence of smaller cells. The blastula consists of many layers of blastomers. Gastrulation. Cells undergo major movements to form the three germ layers. Ectoderm, outer layer, forms skin, nervous system. Mesoderm, middle layer, forms muscles, bones, circulatory system. Endoderm, inner layer, forms the digestive and respiratory systems. Key processes in gastrulation. The vegetal pole. For what is germ layer? Discuss structure and function of extra-embryonic membrane in bird. Answer: germ layers. Germ layers are the primary layers of cells formed during gastrulation in embryonic development. These layers give rise to all the tissues and organs of an organism. Types of germ layers. Ectoderm, outer layer. Forms skin, nervous system, sense organs, and glands. Mesoderm, middle layer. Develops into muscles, bones, circulatory system, kidneys, and reproductive organs. Endoderm, inner layer. Gives rise to the digestive tract, liver, pancreas, and respiratory system. Extraembryonic membranes in birds. Birds develop 4 extraembryonic membranes that support embryonic growth but do not form part of the body. These membranes provide protection, nutrition, and waste management. Yolk sac. Surrounds the yolk, providing nutrition to the embryo. Contains blood vessels that transport nutrients from the yolk to the embryo. Amnion. Forms a fluid-filled sac around the embryo, protecting it from mechanical shocks. Prevents dehydration and provides a stable environment. Chorion. Outermost membrane that aids in gas exchange between the embryo and the environment. Works closely with the allantois to form the chorioallantoic membrane, essential for respiration. Allantois. Acts as a waste storage site for nitrogenous wastes. Helps in respiration and calcium absorption for eggshell formation. These membranes play a crucial role in the survival